So I wanted to take a few minutes today to talk about two different aspects of the course. The first being Twitter. I want to go over it a little bit and just give you guys a little bit of a conversation, a little bit of a pep talk maybe. So first thing is, we're doing really well. So just to give you an idea, we're getting about 4,000 impressions a day in the Twitter account. So this account was set up just for this course. So all the activity on it is associated with this course. So we've had, in the last 24 hours, 3,800 impressions. That's about 28,000 impressions a week that you guys are interacting with course material, which is actually really, really good. So who is having anxiety associated with the Twitter? Lots of people. All right. It is not meant to be a difficult thing. It is not meant to hurt your grade. It is meant to be a different interactive way to get the message out about food and health. So a couple things. First, if you guys are a 90 student on the midterm and final, and you get a 60, let's say, on the Twitter assignments, there is no way in good conscience that I would ever drop anyone a letter grade associated with a Twitter account assignment. So that's first. So, you know, you can reduce a little bit of the anxiety. The other thing is there's an alternative assignment. So I've had a few people, for a couple different reasons, really not want to engage in Twitter. If you are adamantly opposed to Twitter, you can write an essay. The essay is going to be 10 pages. It's going to have to use 10 papers from the article. So exactly the same format as what you would have in most of your other classes. The research has been done with the articles embedded into the course notes. I wouldn't recommend anyone do it. It's going to be a lot more work to write an essay than it's going to be to send 10 tweets. And I'm going to be the one marking the essays. So I'm going to mark language. I'm going to mark, you know, are you able to communicate in complete sentences? Do you have a good flow? I'm going to mark it like I would mark an article. So if you're adamantly opposed for any reason, fire me an email and just say you don't want to do the Twitter assignment. You'd like to do the alternative assignment. And I'll send you a course, like a syllabus type thing for the assignment. Okay? So again, you have an option. There's going to be some other things too for people who are not doing that well in the Twitter. The Twitter space who's not if your grade is low, I'm going to come up with a smaller assignment that you can do to supplement your grade. We're going to talk about that later because we're not in a situation where I want it to happen yet. So we'll talk about that in due course. So again, if you really want to do an alternative assignment, that's fine. If I were to ask a question, so right now, based on Twitter, one question on the midterm would be based on hormonal control. So I wanted to give you an example of the type of questions that are going to be on the final exam. So if I were to ask right now, how would you design a food to increase satiety based on hormones? Who thinks they could put something down? It doesn't have to be a lot. It could be about ghrelin, it could be about gastric emptying, it could be close to kinin. There's tons of different aspects you could talk about. You know, we've talked about it, different hormone controls with neuropeptide Y, with ghrelin, with leptin, with CCK, PPY, PP, PPY. We're going to talk about GLP-1 today. So you don't need to memorize everything, but you need to retain enough that you can actually produce an argument. So the questions aren't going to be like, what are the major hormones in signaling appetite? That's just rote memory. I could take a 7-year-old and I could get them to memorize the different hormones and they would know nothing about what it means, but they could recite the lecture notes to me if I had enough time. What I want to see you guys be able to do is be able to assimilate that knowledge. So we've talked about gastric emptying, we've talked about distension, we've talked about residence time, and all of that is influenced by the amount of fiber. We've talked about that. I want you to start making connections. So by continually going into Twitter and seeing those little tidbits of information, then it's going to continually keep it in your memory, and you're going to find when you come to study for this course that you've constantly been reviewing. The other thing that I did very intentionally was embed those articles into the course notes. That forces you not to forget about them for the semester, which I did, and I would imagine 85% of you do, is you don't look at that course package, that course notes, again until a week or two before the exam. This forces you to go back in to find an article, and by doing that, you are subconsciously learning. You may not recognize that you are, but you are actually acquiring more information every time you go in. So this constant going back to the lecture notes by going to YouTube to watch anything you need repeated, you're constantly getting exposed to that information, which should make studying much, much easier for this course. Everyone's seen that the course notes are going up online. You're seeing the level of detail that you need to know. It's not nearly the same extent that I cover on the course material. What I'm trying to do with the course material is highlight and not dilute the science for you. So when you go into third, fourth year, if you potentially go into a master's program or doctoral program or into medicine, you're going to have the background information, the scientific content for what you learned. Now, when I talked about this course, I said that there's two really cool things that we're going to do. Well, I think they're cool. You guys may not. One I'm not going to tell you about until we're almost there. The other one is, so early this summer, I found out I am the perfect example of metabolic syndrome. So I'm 40 years old. That sounds so bad. So sad when you get old. Enjoy your time now. So I am a prime example for metabolic syndrome. Now, in August, I went on a fad diet. And I've been doing this fad diet now for 33 days. And in 33 days, I've lost 25 pounds. So what I've done is at the end of August, I did a whole bunch of testing. And I kind of did testing in collaboration with a few external labs and a few labs at Guelph and some other, like with my family doctor. So I'm going to show you the changes, like a supersize me type thing, but with this other fad diet. It's not the supersize me diet. But I'm doing a fad diet, which I'm not going to tell you what it is. I'm going to drop clues through the course of the semester. And then we're going to do, at the second last class for the course, we're going to go over all the medical results, and we're going to talk about why those specific markers are important. So the one video, so I'm going to close this for a second. So this might be a little bit end show, tricky for me. And I got to go over. This is an example. Hopefully the sound works. Oh, there is no sound. So that's me. Anyone know what that is I'm in? Do you know what it's doing? So not your cardio, no.
Is it testing like some sort of like ketone bodies through your mouth?
No, any others? So what it's doing right now, that machine, is it's pumping air into the machine and it's finding my percent body fat really accurately. So it's called the bod pod. So it measures the volume, which you are. So by knowing your volume and by knowing your mass and by approximating the density of your bones and all that stuff, it comes up with a percent body fat. I was high. We'll talk about it at the second last class, but I was classified as obese at the beginning, so I'm not anymore, which is good. But we're gonna, at the second last class, so the second last class, we're going to talk about all the different tests I've done, and there's a lot of cool stuff. It's not just going to be blood work. There's some really interesting stuff that pertains to this course. So through the course, I'm going to give you hints on some of the tests I'm doing. So one is the bod pod. There's 4 big ones I'm doing, and they're all different markers of health associated with this course. So we're going to talk about those as a learning, as kind of an example of the techniques and what they actually do. So that'll slowly evolve. You'll find out what the tests were and what the diet was. Okay, let me get back to my lecture slides. There they are. Play from current slide. Not yet. And then I've got to click here, swap displays, and then I've got to swap displays again. Okay, so last day we were in the small intestine talking about different attributes that facilitate the absorption of the macronutrients. So we're talking now glucose, amino acids, and fats. We talked about the sugar transport. So sugars are facilitated transport with the different with the different salts. ATP is then used to transfer the salt out of the cell, and that sugar is taken up by the portal vein. Now, that pickup by the portal vein is extremely important because that portal vein goes directly to the liver, and anything that's in that portal vein or in that fluid is going to be partially metabolized. That's important for when we talk about things like bioactives. So if you're trying to increase the bioaccessibility of a bioactive, let's say vitamin C, vitamin E, whatever it may be, if it's soluble in the aqueous phase and goes into the portal vein, about 30% reduction just in that first pass metabolism. And that first pass metabolism is when that, again, those molecules are picked up by the portal vein, are transferred to the liver, and oxidized and metabolized. So when we talk about macronutrients, it's both the proteins and the carbohydrates which utilize the portal vein. We talked about the formation of the chylomicron, which is when we have the transport of your fatty acid and your monoglyceride, which is then reassimilated in the cell and then transferred out of that cell as a chylomicron with some of the proteins and glycoproteins from that cellular matrix. When the lipids are transported through the cells, they go into the lymphatic system. The lymphatic system allows for systemic circulation. It then goes to the thoracic vein, or the thoracic portal, and transferred into the systemic circulation, which then goes to the liver and metabolism. So that you get a first exposure of what's ever in that fat phase before metabolism happens. So by getting a micronutrient, a vitamin, whatever it is, to be in that fat continuous phase, if you can get it into that lymphatic circulation, you bypass first pass metabolism, and this means that you're going to increase the bioaccessibility of that nutraceutical. That's extremely important. When we talk about, in a couple weeks, we're going to talk about some of the different micronutrients and how low the bioaccessibility is of those micronutrients. Things like ECGC from green tea, resveratrol from wine, lycopene from tomatoes. All of these things that you hear are so good for you have extremely low bioaccessibilities. 5, 10, 15% of what is in that food is actually absorbed. So very low. So any way we can increase the retention or the uptake of those bioactives is extremely good, because the cost for these are extremely high to put into foods. So if you're a food manufacturer and you want to put in one of these novel bioactives, you want to structure them in such a way that you maximize the biological efficacy of those molecules. Does that make sense? What was that? That just beeped. I don't know what it's doing. Okay, so that small intestine is a very undulating, lot of surface area. This allows for first the expansion when food is incorporated, but it also allows for a lot of contact between the vasculature of the human body and the intestine, allowing for greater absorption. We have those microvilli, again, increasing the absorption, and if we go past the villi, we have the epithelial cells, or the microvilli. It's those microvilli that have the brush border, so they produce that mucus to prevent self-digestion, but also this is where absorption happens. So this is where we have the transporters for glucose, fructose, and galactose. This is where we have the transporters also associated with fatty acids. When we talk about the state of the epithelial cells, again, that undulation, or those small microvilli increase the surface area. Now when we talk about the salivary amylase, that breaks down polysaccharides into dextrins, limit dextrins, maltose, and small chains of maltose, things like maltotriose. Now, remember that salivary amylase does not break down into the individual monosaccharides, and the individual monosaccharides are what are absorbed. All of that digestion occurs at the brush border. So there's the 3 enzymes at the brush border. There's sucralase, maltase, and lactase. One of those 3 become dormant as you get older. Anyone know which one of those 3? What foods become harder to tolerate as you get older? You guys still might be lucky. What's that? Sweets. No, sweets? Well, you never tolerate those well. Yeah?
Like
Lactose intolerance, right? As you get older, the need for lactose as an energy source decreases. It's very important when you're a child, when you're breastfeeding, or when you're infant formula feeding. But as you get older, you don't really rely on lactose for an energy source. So a lot of us, our lactase gene turns off and we no longer produce lactase, so we become lactose intolerant. So what happens, because you aren't digesting that lactose, you can't take it across your epithelial cells because it's the disaccharide. So what happens to it? Where does it go? It ends up in the colon and it's fermented. And when it's fermented, it turns into things like methane gas, right? So that gives flatulence. So we'll talk about the different pancreatic structure, the pancreatic lipase in a minute. It's the pancreatic lipase has the same efficiency as the salivary amylase, plays a much, much larger role in carbohydrate digestion. And then we have, whoops, I didn't have it on that slide. Then we have the brush border enzymes. Now, when we have amylopectin, this area of amylopectin where we have these branch points, and we're going to talk about those. So our linear points, all of these bonds are 1,4 alpha glycosidic bonds. We have the enzymes that are able to break these down, these bonds. We don't have the enzymes associated to break the 1,6 alpha bonds. So those reduce the glycemic index of a carbohydrate because of the formation of limit dextrins, right? So the limit dextrin is about 2 or 3 carbohydrates away from that glycosidic bond, and we can't break those down. We have to use a different set of enzymes, which are far less efficient. So again, when we have foods high in amylopectin versus amylose, the glycemic index drops because the rate at which glucose is produced and absorbed decreases. That makes sense. Everyone kind of understands that? The pancreatic amylase, those small dextrins and disaccharides move to the brush border. They can diffuse through that mucus layer, and they're interacting then with the 3 brush border enzymes, which then cleave the disaccharides into monosaccharides, which are then available to be absorbed, transported through the epithelial cell to the portal vein to the liver, first pass metabolism. Again, the 3 main enzymes at the brush border for carbohydrates are your maltase, your isomaltase or sucrase, and your lactase. Again, the lactase decreases as age progresses. This is also dependent somewhat on race. From an evolutionary perspective, how long dairy products are a major component of your diet. So geographically, this is highly variable. There are some cultures that drink large amounts of milk late into life, so they continually produce the lactase gene. If you're weaned off milk at a fairly young age, you're more likely to produce less lactase. So we've already talked about the surface of the microvilli. We have 2 main transporters in. One transporter works with glucose and galactose. The second transporter works for fructose. Now, once they're in the cell, it's the same transporter that transports them out of the cell into the portal vein. Now, when we talk about the glycemic index is the rate at which, or the amount of glucose, be careful, it looks like you have a whole bunch of water under you when you get up, just so you don't fall. Sorry, I got distracted. So the glycemic index is the area under the curve of your glucose profile, right? So it's the rate at which glucose increases in your blood. Now, what about fructose? What about galactose? Do those affect the glycemic index? Because again, it's not glucose. What happens to those compounds? Well, once the fructose and galactose enter the portal vein, they're transferred to the liver and they're converted. And there's a rise then associated with these, and we're going to talk about this a lot in the carbohydrate section, that fructose and galactose can be converted to glucose, but at a much, much slower rate. So instead of having this rapid increase in blood glucose, which is detrimental when we talk about an insulin response, but when we see that rapid increase, fructose slows that down because fructose is absorbed, sent to the liver, converted to glucose. So that, again, that glucose profile changes depending on the composition of the monosaccharides that we are absorbing. That was strange. We talked about lipid digestion. Remember the role of CCK, so cholecystokinin. It triggers the gallbladder to contract. The contraction of that gallbladder secretes bile into the intestinal phase, and that bile is made up of cholesterol. The difference between cholesterol and bile is the 2 extra hydroxyl groups that are on bile. Now, why does that matter? Again, we have that aromatic, that cyclic structure of carbon. We have those hydroxyl groups that extend downward. The downward profile of that is very, very hydrophilic. It's going to choose to reside in the aqueous phase. That ring structure is all carbon. It interacts with the oil phase. That then reduces the interfacial tension between those 2 phases. We're going to talk about why this is important when we get to lipids, but it reduces that interfacial tension between those 2 phases. And when your stomach undergoes contractions, when your intestinal compartment mixes the food, it's reducing the particle size. The reduction in particle size allows for a higher increase in the surface area of those droplets. That is your substrate of your reaction. The enzyme is in the aqueous phase. So we need that high surface area so that that lipase can then interact at the interface and digest the substrate triglycerides into the fatty acids, the 2 fatty acids, and the 2 monoglyceride. So the 2 monoglyceride has that fatty acid at position 2. Glycerol has 3 carbons, 3 hydroxyl groups. Those hydroxyl groups can form a glycosidic bond, not a glycosidic, the name of it's gone, I'll get it in a while. So there's 3 fatty acids on glycerol. It can remove the SN1, SN2 position, SN3 position. It cannot remove the SN2 position. So we absorb 2 fatty acids and 1 2-monoglyceride. These then are conjugated, the bile, to either glycine or toluene. These make them more water soluble. Again, that bile goes to the interface. It interacts at the interface, reducing that interfacial tension. That allows then lipase and colipase to come to that interface. Both are required for pancreatic lipase. Remember, maternal lipase and lingual lipase do not require bile. Pancreatic lipase requires bile to work. If bile is not present, pancreatic lipase efficiency drops way, way down. So the pancreatic lipase, again, is the responsible enzyme for lipid digestion in healthy adults. We absorb the fatty acids, we absorb the 2-monoglyceride. Those are reassimilated into that cell, into the lipid, the triglyceride. Those triglycerides are then incorporated into chylomicrons, so it's a lipid protein. And those then are picked up by the lymphatic system and go through systemic circulation. Now, something I want you to start thinking about. Everyone understands glycemic index of foods. Glycemic index is this term that we use and we talk about foods all the time. We make choices based on the glycemic index. What about a lipemic index? Does the rate in which a fatty acid enter the bloodstream important? Evidence is starting to suggest that it may play as big of a role as glucose in establishing as a marker for health. So if you eat a fat that's readily digested and absorbed, could the rate in which those fatty acids absorb affect health? So is there such a thing, or is there a need for a lipemic index, just as there is for a glycemic index? And we'll talk about that. So with the lymphatic system, again, we're taking up all the fat-soluble nutrients. The chylomicrons are going into the lymphatic system. This typically isn't controlled. There's not a lot of control over that. Now, the benefit of this is that fat bypasses the liver. So again, it bypasses first-pass metabolism. It's got to get to the thoracic duct and then through systemic circulation and then to the liver before it's starting to be oxidized. So lipids, and especially when we talk about fat-soluble compounds, we can really increase the bioaccessibility by bypassing that first-pass metabolism. The other thing that the food industry is starting to do is say, okay, the caloric density of a triglyceride is higher depending on the chain length of the fatty acids that are associated with that fat. So the chemical structure of a fat changes how much energy you're going to get out of the fat. depending on the chain length of it. So what we're starting to think of doing is what happens when you put really, really short chain fatty acids. So fatty acids are carboxylic acid group with a long aliphatic chain. That aliphatic chain length changes depending on its structure, depending on its source. So if we have an animal fats are mainly made up of stearic acid, butter, lard, tallow. It is made up of mainly stearic acid, palmitic acid. So stearic acid has 18 carbons, palmitic acid has 16 carbons, or oleic acid, which is 18 carbons with one double bond. If we don't eat animal-based products, instead we're going to rely more on the palm oils, which are then used to structure and make solid fats. Palm oils are more of your medium chain triglycerides. Things like palmitic acid, so C16, lauric acid, musteric acid, so C12 and 14, are the big ones in your palm kernel oil. Those fats have different effects on your HDL to LDL ratio. We're going to talk a lot about this in the lipid section. So depending on the source of your fat, it can change the healthfulness of those metabolites. Why is this important? Right now, does anyone know what legislative requirement went into effect in the last 2 days? Yes. So we can't put trans fats in foods. We're going to talk a lot about trans fats later on. But what or why did we invent trans fats? Does anyone know? Yeah. In some instances, yes. Yeah, to increase shelf life because unsaturated oils oxidize much more readily. Yeah.
Solidify like oils and stuff that needs to be solid.
So it'll convert a liquid oil, something like canola, sunflower, safflower oil, into a solid fat like butter. This all happened in the 70s and 80s because there were some reports that came out that said saturated fats increase the incidence of coronary vascular disease. This has changed the opinion recently. We'll talk about it later. It's a little bit too much for right now. But what I want you to know is that the chain length of that fat changes its biological fate. So if you incorporate short chain, C2, acetate, C3, propanoate, C4, butyrate. Anyone know where butyrate is found? Really common in one food, in dairy products. So if you ever smell the smell of milk, that's butyric acid, one of the things you're smelling. Those can actually be utilized by the cells, the epithelial cells, as an energy source. So they don't even make it out into the lymphatic system. They're incorporated and used as a building product for metabolism within those epithelial cells. So when we talk about the short chain fatty acids, if we increase the amount of short chain fatty acid within a triglyceride, we're going to decrease the caloric content of that fat. So we can restructure, we could add a whole bunch of butyric acid, incorporate it onto the triglycerides, and all of a sudden we've decreased the caloric density of that food. Because your body responds differently to the short chain fatty acids as it does to the longer chain fatty acids. And we'll talk a lot about this in a few weeks when we talk about lipids. So at the end of the small intestine, again, the nutrients are predominantly digested and absorbed. We now have another hormone that's released at the end of the ileum, at the beginning of the large intestine. And this is glycogen-like peptide 1. Again, like all of the hormones associated with digestion, they play a role in appetite suppression. They also trigger now the insulin. They trigger the pancreas to start producing insulin, right? So at this point, when the food reaches the ileum, most of the absorption has taken place. If most of the absorption has taken place, our blood sugar is going to be elevated. If our blood sugar is elevated, our biological response is to produce insulin to get the other tissues in the body to pick up that glucose. So glycogen-like peptide 1 triggers insulin to start being produced to reduce the amount of free sugar in the blood. It facilitates the uptake of glucose by the muscle tissue. it decreases, it tells the liver to stop producing glucose. You don't need to metabolize any more glycogen at this point. We can rely on the blood sugar the sugar that is in our blood at this point. Insulin also plays a role now. So glycogen peptide 1 triggers the release of insulin. That insulin also has roles in appetite suppression. It also slows down gastric emptying, and it starts to tell the stomach there's no more food coming. Let's turn down the HCl so we don't start to damage that gastric tissue. So this whole concept of hormones interacting with satiety is what we call the ileal brake. The ileal brake of a food determines how satiating it is. Do you feel full for a long time? This has to do with our neuropeptide family, so the neuropeptide Y, PPY, PP as well. Oh, that sounds really weird. We've got our gastric emptying rates. When we talk about gastric emptying rates, we're talking about ghrelin, we're talking about the hunger hormone. In cholecystokinin, we're talking now about gastric motility, secretion of enzymes, and we have the glycogen-like peptide 1 from the intestine. So all these hormones stimulate appetite suppression. So this ileal brake is how these foods trigger these hormones. And if you can design a food to affect the ileal brake, you can begin to tailor the satiation of that food. This becomes extremely important. This becomes important in things like if you're treating an HIV patient in a third world country that doesn't have access to a lot of nutritional resources, you've got to maximize the caloric density of that food. You want them to feel hungry so they consume more. So when you start talking about designing meals for people in developing countries with HIV, you need to think about the ileal brake. You need to think about satiation. You need to think about the fact that these people's appetite are suppressed as it is. So if they have a suppressed appetite and you're giving them a food that further suppresses appetite, it's going to have bad biological effects. So we can tailor foods based on the needs of the consumer. If you're an athlete, if you're a bodybuilder and you're consuming 6,000 or 7,000 calories a day, you want to eat your meal and feel hungry again. You want to be able to consume a lot of calories. So again, when you design a whey protein or a supplement that you're going to use for marathon runners, again, all of that changes. The way we want our body to interact with that food is dependent on what you're doing. This again introduces that whole concept of personalized nutrition. Someone like me doesn't need that same effects of food as someone that's going to go run a 24-hour marathon, an ultra marathon. I certainly don't need the same number of calories, although I probably consume more than them. So we've talked about the glycemic index and how the structure of that carbohydrate changes the glycemic index of that food. We're going to talk about at a later point how the structure of the food changes the digestibility of it. So again, you can think about it. If you have a really dense particle of food versus a liquid food, the enzymes are going to be able to much more readily access the interface or the substrate and break it down. So we can design foods to have different glycemic indexes. Can we do the same with triglycerides? Again, we'll talk about this later. So does the structure of the crystals in fat, ooh, I'm going through puberty apparently. Do the structures of fat, the size of the crystal, the shape of the crystal, what we call the fractal dimension of the crystal, so how it's arranged in space, if it's a highly branched network, that all affects the rate at which lipase can interact with that substrate. If we're changing the structure, we change the lipemic index, and we can again begin to tailor the structure of the foods to have a distinct nutritional profile. We can engineer exactly what we want based on the chemical structure of that food. But here's the scary thing. When we process foods, when we refine them, when we homogenize them, when we manipulate the native structures in foods, we typically increase the digestibility. So now when we talk about things like insulin response to sugars, if the rate in which that sugar is absorbed differs than what we have been consuming for hundreds of thousands of years from our evolutionary perspective, can our insulin keep up? Is that enzyme tailored to be able to reduce the glucose in the body if it spikes? No. We know that's not the case. That's why so many people have type 2 diabetes, is because all of a sudden we're producing foods that are so digestible, that have so much sugar, that our insulin mechanism can't keep up. So we oversecrete insulin. For years and years and years we do it. And then all of a sudden our cells say, you know what? I've been so overexposed to you. It's like a really bad girlfriend, in my case. You want to get rid of it. You don't want to interact with it. It becomes less effective, right? That insulin can't stimulate the cells to pick up glucose. And we develop type 2 diabetes. Millions and millions and millions of Canadians and Americans, well, anywhere in the Western society, is starting to develop this. The challenge with it and the real problem with it is these diseases are managed for very long periods of time. So you endure the cost of my metabolic syndrome. Your taxes pay for my medicine. So this becomes a huge problem. And it becomes an even bigger problem when our medical resources, as it is in Canada, are limited. So we have a very finite number of doctors, a very finite amount of equipment that we can test people with. When there's people like me burdening the healthcare system, it's people like you that suffer. So we've got to start to think about how we can design foods, how can we process foods, so that they don't change our biological makeup, so they don't induce disease. Again, estimates as high as 65% of the adult population in Western societies is affected with a diet-related diseases. This increases as your socioeconomic status decreases. So the poorer you are, the more likely you are to eat highly refined, highly processed foods. This increases the likelihood of you developing diseases. So when we talk about metabolic syndrome, when we talk about type 2 diabetes, these afflictions are far more likely to affect you if you are from a lower socioeconomic status than someone else. So it's something to think about. The whole idea of the lipemic index is all being fostered here at Guelph. There's about 10 of us that are working on it. Amanda Wright leads the way. So any nutrition students that are in the non-dietetics program will know Amanda Wright, or should know Amanda Wright. Amanda Wright runs the Human Health and Nutraceutical Science Research Unit. So if you ever do a feed and bleed study, so you go and eat something, they stick a needle in you for a couple hours and measure the amounts of nutrients going into your blood, Amanda's the one that's going to do it. Or Dr. Wright. Dr. Wright was also my PhD co-advisor. She's now doing a study. Now just think about the basics of this. The size of an emulsion droplet will affect the rate in which you would digest that fat. What foods do we homogenize? Anything that has fat in it. Any beverage that has oil in it. So think about things like salad dressing, mayonnaise, any sauce that you use. We very closely control the droplet size. Why do we homogenize milk? We homogenize milk to increase the shelf stability of that milk. Why is it increasing the shelf stability of that milk? Because we're decreasing the particle size. And we'll talk about Stokes' law when we get to lipids. So Stokes' law is the rate of sedimentation, flocculation, creaming, how fast it separates. So if you don't homogenize milk, if you go to the farm and get fresh milk, it's going to cream in a matter of hours. You're going to leave that glass of milk on your counter. Very quickly, you're going to get a fat phase. You're going to get a serum phase. We homogenize it. We decrease the particle size. That decrease in particle size increases the stability. So from a food chemistry perspective, it's great. I can make an emulsion that's going to exist for years. When you go buy a salad dressing, when you go buy mayonnaise, they exist as mayonnaise. They don't exist as a water phase and an oil phase. Again, this all has to do with the droplet size. Now, if we decrease that droplet size, we increase the number of particles that are smaller. By doing so, we increase the surface area. If we increase the surface area between the substrate and the enzyme, we're going to increase the rate of lipolysis. We're going to increase the rate in which those fatty acids enter your bloodstream. We don't know the biological consequences of that yet. We know it happens, we know we can tailor it, but we don't know how to control it yet, and we don't know what the long-term biological effects is. And the challenge is within the food industry is you're not going to get a toxicity effect from a single exposure. What about from a chronic exposure? From a lifetime of eating highly processed, refined foods? The chronic effects will magnify. Will they play a role in developing type 2 diabetes and metabolic syndrome? Likely. Evidence suggests that it will. So how can we make an emulsion with a large surface area, or can we structure that interface in such a way that we limit lipolysis? All of these things is what the food industry is looking at now. How can we tailor it? So now we're going to talk about protein digestion. The vast majority of protein digestion occurs in the intestinal phase. We've already talked about the gastric effects. So we have first that exposure to the low pH. That low acidification denatures that protein, causing the protein to unfold. By unfolding it, we're again increasing the surface area between the substrate, the protein, and the enzyme. So that all happens in the stomach. It then goes into the intestine. CCK triggers the release of bicarbonate to neutralize, bring that pH back around 7. And then we get two main proteolytic enzymes. Those main proteolytic enzymes break down amino acids into bioactive peptides. Bioactive peptides will confer some kind of biological response. They've been shown to do things like mediate blood pressure. So your ACE inhibitors, your ACE inhibitors, those will change your blood pressure. Dairy products are digested into bioactive peptides. And then we have the amino acids. The amino acids can be absorbed, transferred through to the portal vein, to the liver, undergo assimilation, and rebuild peptides or proteins. Now, the two main enzymes that are secreted, again, are zymogens. Zymogens are the inactive form of that protein, so these aren't going to digest proteins themselves. Again, why is that important? Because if a cell produced a proteolytic enzyme, you would self-digest yourself. So we produce trypsinogen and chymotrypsinogen. Now, the difference between these two is the amino acids in which they interact with. So when they cut that peptide backbone, it's going to depend on what amino acids are there and allows those zymogens or enzymes to interact with. So both of these are secreted in the duodenum. The trypsinogen interacts with a brush border enzyme called enterokinase. This changes the zymogen, trypsinogen, into the active enzyme trypsin. Trypsin then is in the compartment. It will interact with it'll self-digest, so it'll be able to activate itself, but it's also the only way we can activate chymotrypsin to begin to work. And again, the substrate specificity differs depending on which protease you use. So if you have trypsin, it's going to act mainly on lysine and arginine. If you have chymotrypsin, it's going to act on your large hydrophobic, typically aromatic, so your phenylalanine, tyrosine, and tryptophan. And again, there's your aspartic acid, your histidine, your serine, your catalytic triad that allows that attack of that carbonyl group. Again, evolution has perfected these enzymes. Okay, Nawaz, do you want to start taking these down the middle and just passing them down the sides? I don't want you to eat them yet. So rennet. Rennet is an enzyme that's found in the stomach lining of calves. So in the 1960s, all cheese was made as a byproduct of the veal industry. So as the cow ages, the reliance on chymosin decreases, and other proteases are produced. Chymosin is remarkable. it only interacts with one peptide bond. So the specificity of that enzyme is highly, highly tailored. So in older cattle, or in older calves, we switch from mainly chymosin being produced, which is again highly specific, we're going to talk about that in a second, to pepsin. Pepsin is not that specific. Don't eat them yet. We're gonna watch a video before we eat them, which is really cool. So we don't have a large enough veal industry anymore, which is probably a good thing, to support the cheese industry. So there's two alternatives. First, there's vegetable-based rennets for vegetarians. Has anyone ever eaten a non-animal cheese? Or, yeah? What would you describe the texture like? Sticky. The texture, because vegetable-based rennets are not specific to this one bond, which we're going to talk about in a second. Chymosin is. So where does chymosin come from now, or where does rennet come from? It is all recombinant. So if you are a hardline anti-GMO person, no cheese for you. Almost all cheese now is made with recombinant E. coli rennet. So if you really are an anti-GMO person, there are very few cheeses you can actually eat. And most of the cheese quality, if you're not eating recombinant, is very low quality if you're relying on vegetable proteases. So we've got to take a little bit of a sidestep here. We're back to milk. So milk has two main proteins, right? You've got your whey proteins and your casein proteins. Whey proteins are now mainly used in the supplement industry. Caseins are mainly made into cheese. So it's the casein micelle that is designed remarkably to make cheese. So the casein micelle is an aggregate of hundreds of proteins. So it's this large spherical object. Now, this image has been cited more than 350 times. That's remarkable for a single image. And this was taken by Dr. Paul Spagnuolo, who's now a professor in the Department of Food Science here at Guelph. We just took him from the University of Waterloo Department of Pharmacy. And why is that over there? Sorry. So the casein micelle. So the casein micelle has four main proteins. We're going to talk about this a lot in the protein section. It's got alpha S1, alpha S2, kappa casein, and I forget the fourth one. But those four proteins, depending on the polarity of them, might interact in this micelle in different ways. So three of the proteins reside in the core. They're very hydrophobic. They have a lot of sulfur groups. So those reside in the core of that micelle. Now there's something else that exists in the core of the micelle. Calcium phosphate. Why is that important? The solubility of calcium in any form, in any salt, calcium chloride is the highest, but still, it has a much lower solubility threshold than casein in milk. So that means we can get more casein in the casein micelle in milk than we could get calcium in a solution of water. So the casein micelle is designed to be a delivery vesicle for calcium. So three of these proteins are very hydrophobic. They need to reside in that core. They don't want to be exposed to water. One of these proteins, and it's kappa casein. Kappa casein, so we've got alpha S1, alpha S2, and beta casein. Those reside in the core. We've got calcium phosphate, which is embedded within that protein matrix, increasing the amount of calcium that can be delivered in dairy products. And then we've got the kappa casein. Now the kappa casein is important because it carries a negative charge. Why is that important? If two casein micelles come in close proximity, we get electrostatic repulsion, right? So this kappa casein has a hydrophobic end and a hydrophilic end. So that hydrophobic end resides within that protein matrix and it extends out what we call a hairy layer. And that hairy layer is water soluble and negatively charged. So we decrease the interfacial tension between the proteins and the water, so they exist in solution, but we get that electrostatic repulsion that keeps them in solution. Now, kappa casein in its protein backbone has this 105, 106 phenylalanine methionine bond. That's the only bond chymosin can cleave. The only bond chymosin can cleave is that specific 105, 106 of kappa casein. It does nothing else. That's it. Now, why is that important? And this again shows how remarkable evolution is. We design a single protein, a single hormone or enzyme, to digest one single bond. One bond found in milk. What does this do? So this cleaves the para-kappa casein from the glycomacropeptide. The glycomacropeptide is water soluble. So when you cleave that, it floats away into the aqueous phase. I saw you eating the cheese. The para-kappa casein is very hydrophobic. It stays with the casein micelle. So when you begin to trim off that hairy layer, you begin to reduce the electrostatic repulsion, so two casein micelles can now interact with each other. The hydrophobic core is now exposed to water. It becomes insoluble. So now what happens is we have this exposed para-kappa casein. All of the electrostatic repulsion is gone in this area. All of the stability imparted by that glycomacropeptide is lost. It's insoluble. It's got to find another casein micelle to interact with to prevent that exposure of that hydrophobic protein from the aqueous phase. So here's a mixture of cheese. So we've got your casein micelle. You've got your whey proteins. So your whey proteins aren't typically used in most cheese manufacturing, except for ricotta cheese. Ricotta cheese is one of the only cheeses that is made by using whey protein. Everything else is made by casein. So what happens? You cleave that hairy layer off, and all of a sudden you get aggregation. You get the formation of cheese. That's what you're exposed to. So when you're an infant, or when your cow is eating or consuming milk, it's turning it into cheese. It's coagulating it. It's turning it from a liquid to a solid. Why is that important? Distension, gastric emptying, ghrelin, the rate in which it goes into your intestines, the surface area to allow that proteolysis to happen. All of that changes. It's all tailored to slow down the rate in which it moves through the gastric compartment so you can maximize digestion. Again, evolution has created a single enzyme that cleaves one bond in one substrate just for this purpose. All right. So while you eat your curds,
this one is fresh, not cooled. We have minimal acid development after the curd was salted and removed from the vat. This one has been chilled. Not sure how long, but chilled. Therefore, the texture is much different. There's also more acid development here than here. And we think of cheese curd as very springy. When you put some in your mouth, it's very squeaky. We're going to try to demonstrate that. Listen up.
With the fresh curd, the moisture is still on the surface, and you feel that bouncy, springy texture in your mouth, and you definitely hear the squeak.
Compared to this one, where it feels a lot firmer, no spring to it at all. It's more gummy.
Much more dense.
More dense, yes. It has no squeak. Kind of sticks to your teeth a little bit. Just night and day difference. Now, if you warm this, like right now it's about 5 degrees Celsius. If you warm it back up, so it's over 20 degrees, it will get some of that squeak back. It's never going to be as nice as this. Again, it was that nice once. It'll never be as nice as the fresh product, but it will get a lot of the squeak back. So what's the difference? There are two principal things that will determine the squeak and the elasticity, the springiness of the fresh curd. The first is the temperature. So at warmer temperatures, the protein functionality is quite different than it is at cold temperatures. The other one is the amount of acidity. So when cheese is made, we ferment the milk with lactic acid bacteria, and those bacteria produce acidity. There is an optimum range of acidity as measured by pH. It's about pH 5.0 to pH 5.2. So that's a measure of acidity. There's an optimum range in which the acidity in the curd is just right to produce that springy, elastic type texture. If one of those two things changes, that is, the temperature goes down, so I'm well below 20 degrees Celsius, or with time, the bacteria continue to produce acid, so the bacteria here have also reduced the pH or increased the acidity to a much greater level than it is here. Those two things will reduce the squeak, the springiness, and it breaks easier, and it's a very different texture in your mouth.
So it's not unique anymore, it's not enjoyable, it's not what makes cheese curds so special. So certainly a difference when you can enjoy them fresh.
So think about it for a second here. So we have a dairy product that we've modified using our enzyme, rennet, and depending on the temperature to which we store it, changes how that network forms, which changes your sensory experience. So the chemistry of that system determines that sensory attribute. That squeakiness is a determinant of the quality of the cheese. So when you go to places like Zehrs and you buy curd, that curd's a few days old. You will not get the squeak in that curd. So you won't feel that same consistency that you do in fresh cheese curd. And again, when we talk about food science research, these are the types of things that we try to fix. So can we make a cheese curd that's stable for 4 weeks at refrigerated temperature that remains squeaky? So again, there's so much to food science that we take for granted because that sensory perception, that oral food processing, determines your likelihood of whether you're going to buy that product again from the grocery store. So again, it's remarkable the interactions between structure of foods and the sensory perception of those. So now when we talk about how we're digesting, so now you've got that cheese in your stomach, pH isn't going to do a lot to it because it's already at a low pH, but your proteases begin to interact. So you get your pancreatic protease, which is going to start breaking it down into oligopeptides. Whoops, what happened there? That's going to break it down into shorter peptides. Some of them are going to have biological effects. They're going to be able to interact with different hormone receptors. The amino acids and very, very small peptides are able to be absorbed. Up to 3 amino acids. Anything greater than 3 amino acids will not be absorbed. So again, that brush border plays a big role in breaking down those small peptide chains into single, di, or tri-peptides. Those are transported again, similar mechanism as we see with glucose, relying on sodium. Again, we require ATP to kick that sodium out. They go into the portal vein and off to the liver for first pass metabolism. So now we hit the large intestine. And the large intestine is mainly where water absorption happens. We also there begin to form the feces, but we also get a lot of microbial fermentation. This becomes very, very important when we talk about non-digestible carbohydrates or non-glycemic carbohydrates. Things like cellulose, alginates, pectin, all of these different polysaccharides. And again, we're going to talk about all the different uses, why we use them, and the health effects much later. But the colon plays a very important role in utilizing those non-glycemic carbohydrates. So the microflora in your gut can take food that we cannot interact with, and it can begin to metabolize them. So the bacteria take non-digestible food components, metabolize them into, and their metabolites are things that we can utilize. So one big one when we talk about the non-glycemic carbohydrates is the formation of short-chain fatty acids. So the bacteria can convert non-digestible carbohydrates into short-chain fatty acids. These are used as an energy source in the epithelial, so they have a huge role in preventing colon cancers, but they also produce enzymes for different bonds that our body can't break down. This is great when we talk about, again, the non-digestible carbohydrates, but it can become very problematic. What happens if we take a compound or a food that's safe, completely safe to eat, and is metabolized by a bacteria into something that's carcinogenic? Happens all the time. So we'll talk about that in a second. So when we talk about the microbial effects of the colon, if you have a healthy gut microflora, it's excellent. It produces a lot of metabolites that you can utilize for energy, but you can get in dysbiosis pretty quickly, or when that population shifts. Everyone's starting to hear in the news about C. difficile infections in hospital. These are bacteria that can colonize the colon and begin having very detrimental effects. So even though there may be some great effects from the microorganisms, there can be some very dangerous effects. And again, when we get to that idea of personalized nutrition, when you think your gut microflora consists of anywhere between 500 and 1,000 different species of bug, and each of those bugs produce a different set of enzymes that can metabolize different foods, we've got to start to think that the idea of personalized nutrition can have a huge impact on the gut microflora. So again, non-glycemic carbohydrates can be digested into short-chain fatty acids. So we can also have non-digestible carbohydrates bind bile. If it binds bile and exits in the feces, we need to create more bile. How are we going to do that? We're going to utilize cholesterol. So if we are utilizing cholesterol to make bile, we're actually going to reduce the cholesterol in our body. And what's remarkable and amazing is this gut microflora is established within a few days of birth. So you are born, I've got to choose my words very carefully. I almost said you're born sterile. You're born with no gut microflora. So when you're an infant, if you were born by cesarean section or vaginally, you are going to have a distinctly different population. So long-term health effects of cesarean sections, again, when they modify the gut microflora, what is that biological effect? We don't know. If you were bottle-fed or breastfed, significantly changes the population of bugs in your colon. What are the long-term effects of that? We don't know. Gestational age. So how long were you in your mother? Again, is going to change your gut microflora. If you were born in a hospital, if you had antibiotic use as an infant, all of these things will change your population of gut microflora into your adult life. Why is this important? We'll talk about it in a second. So again, if we look at a phylogenetic tree, so each line represents a different species of bacteria. Based on this study, the average person has around 400 different species of microorganisms in their colon. So now we talk about personalized nutrition, and that every person produces enzymes at different rates, and we interact differently with our hormones. But now, each of you are going to have a different population and different ratios of these bugs in your colon. So think about how complex the idea or the concept of personalized nutrition becomes. The population changes based on your age. So we talk about, I can't say these and I apologize again, the spermicides and the bacteriocides. The ratio of these are a very important marker in health, which we'll talk about much later in this course. But that ratio changes in response to age. So a box and whisker plot, the middle of that box is the mean. The box itself contains 50% of the values. and then we have our 25th and 75th percentile. All we really need to look at is the means. So look at our ratio of firmicides to bacteriocytes in an infant near 0, goes to a 10-fold change, and then back to 0 in elders. What effect does that have in how we interact with our food? A lot. Also, the ratio of those two bacteria change depending on if you're obese or not. So your gut microflora changes in response to weight. Now, is it that you're eating different foods when you're obese? It could be. It could just be that you have more calories, you change the species that go, but as you lose weight, you can see your abundance of bactericides goes down. Again, these are important markers for health. And what's remarkable, and what we totally don't understand yet, is if you think about it, we have 400 different species on average in our colon. If we get a 1% or a 2% increase of a bacteria that's been implicated in things like colon cancer, in ulcer formation, we can get into disease states very quickly. But now we look at the metabolites. That metabolite can be absorbed. What happens when it has neurological effects? Could it be implicated in things like Alzheimer's? In things like, I'm trying to think of the other, autism is the other one. So there's now links that show a child with autism has a significantly different population of microflora compared to a healthy child. So imagine for a second if the root cause of autism was based on the microflora that was present in your gut. Think about how easily this could be treated. If we could identify a species that's overproduced in a population of autistic children, could we treat autism with antibiotics? This is where research is going. There's links, there's definitely ties between populations and their gut microflora. So can the bacteria in your colon change mood, behavior, appetite? We already talked about how it can interact with the PYY hormone receptors. So now, could obesity be partly caused by the population in your colon? So what came first, the chicken or the egg? Does our population change because we got fat? Or do we get fat because our population changed? These are the types of questions that we need to answer. Now, we've already talked about the metabolites potentially having detrimental effects. There's also great effects. The production of short-chain fatty acids is extremely important when we talk about colon carcinogenesis. So we know that your gut microflora producing short-chain fatty acids is a good prediction of whether or not you're going to develop colon cancer later in life. So we want to produce short-chain fatty acids. We want people to consume non-glycemic carbohydrates so they produce the volatile short-chain fatty acids, so they can be used by the epithelial cells for energy and assimilation, so they can repair DNA to prevent cancer. Again, we only produce a very limited amount of enzymes that are capable of metabolizing foods. Bacteria have all different ones. And I'm just pointing, you don't have to know this, but there's this set of enzymes called your azeo-reductases. And these are able to break N double bond N. Where are these found in foods? Anyone know? Artificial colors. Allure red. Tartrazine. What happens? We can do all the cell line work in the world, and they're not going to have a detrimental biological effect. You expose them to an azoreductase that's in your gut microflora, it's going to break down into two small compounds. This one here, this one on this side, this one is a potent carcinogen, methyl red. So used in all kinds of cosmetics that everyone will be using. Methyl red is an azodie that's broken down, again, completely safe to eat, but if you have a bacterial species that produces an azoreductase, it is going to turn that dye from a benign, non-biologically significant molecule into a very well-established carcinogen. That's only what we know about. Imagine again the interactions with autism, with inflammatory bowel disease. We have no idea what molecules are being created by the gut microflora because it's so diverse. And there's so many different food constituents that we just haven't had the time to research. Again, it took 12,000 years to figure out that your tooth size responds to cooking. How long is it going to take us to figure out how dyes, how pigments, how artificial flavors are broken down? We know we don't metabolize them. What about the bugs? Do the bugs metabolize them and produce carcinogens that you guys are going to be chronically exposed to for the next 60 years? Ingredient, safe, but its metabolites are very, very different. So this is all stuff we need to think about. So we'll stop here. Take cheese curds on the way out if you want some, please.
Hi. It was really good. And I'll just